immunosuppressant for the prevention and treatment of graft versus host disease. To give you a little bit of background, graft versus host disease is one of the major complications in bone marrow transplant where the donor T cells recognize the patient's tissues as foreign and try to reject it. So it's the one type of transplant where we have to worry about the graft rejecting the patient and not just the other way around, the patient rejecting the graft. In graft versus host disease, its manifestations are most commonly associated with skin rashes that can be quite severe, including bullous formation, and that would be a stage 4 graft versus host disease of the skin. It could also attack the liver, causing liver failure, or attack the gut, causing liters of diarrhea per day, and it could be a life-threatening event. Each of those manifestations are associated with profound immune deficiency, and it's frequently infection that causes the death of the recipient. Now, the mainstay of treatment prevention is calcineurin inhibitors such as cyclosporin A or FK506 or tacrolimus. But there's other new drugs out there that are being used that may be more potent and may be more effective in prevention. However, we're looking at a novel strategy here that Bruce Blazer and Keli Hippen and I and others have been working on for quite a while, and that is taking advantage of nature's way of controlling the immune response, that is the regulatory T cell. And this is a cell that is quite important in the context of autoimmunity as well as in the way of controlling graft versus host disease. So Keli, thank you for coming today to talk about the translational research. And I think that this story and behind all this serves as a great example of PhDs working in a clinical department to help us solve some of the major challenges within the fields that we're working in. And clearly it's a team approach, and Keli leads this effort in developing regulatory T cells for human use. Thank you, Keli.
Thanks, John. So I'm happy to have the opportunity today to move this down a little bit to present our data on 3 different first-in-human clinical trials that we've developed with in vitro expanded regulatory T cells. And I hope to have time at the end to show you our efforts on generating what the next wave of T Reg therapeutics will be. And I would appreciate any questions you might have. We have a small group today, and I've built in a little bit of time. So if you have questions, please go ahead and ask. And if you don't mind, if you raise your hand, I might not see you. So if you want to just speak up, that would be great. And I have no financial relationships to disclose. And so as John was saying, allogeneic stem cell transplantation is an effective approach for treating leukemia, and we have 5-year survival rates well over 50%. The stem cells that I'll discuss today come from either umbilical cord blood or match-related donors. And again, as John was saying, the graft versus tumor effect is driven by host damage activating host antigen presenting cells, and then these host antigen presenting cells are recognized by donor T cells, which become activated and expand. And then these activated donor T cells are able to either directly kill tumor, or they can prime NK cells or monocytes to cause tumor killing. However, this same pathway that leads to tumor killing can also result in host tissue destruction. And as John was saying, the common target organs include skin, liver, and gut. And this severe graft versus host disease is seen in over 50% of patients, and it can cause extensive pathology and even death. So the mechanism and the pathology that's seen with graft versus host disease is very similar to what's observed in autoimmune diseases insofar as you have tissue damage that primes an inappropriate T cell response that leads to T cell activation, priming of the full immune response, and then pathology of specific organs. Now, most of us don't develop autoimmune disease even though we have self-reactive cells present in our body. And this is because there's a subset of T cells called regulatory T cells that normally prevent activation of self-reactive T cells in healthy individuals. So therefore, our hypothesis was that we could use the T Regs also to ameliorate GVHD. So just a little bit of background on T Regs and T cells in general. So T cells develop in the thymus, hence the T, and they use gene rearrangement to produce an antigen-specific receptor. And so T cells with high or no affinity for self-peptide MHC undergo apoptosis. T cells with low affinity for self-peptide MHC are selected into the periphery and monitored for pathogens. And the reason that they need to have low affinity for self-antigen is because they have to have some basal recognition of the antigen-presenting cell. So if they have, no sense of self, they can't be selected and go into the periphery again. And once they're in the periphery, they monitor for pathogens and again become activated when they meet an antigen-presenting cell presenting their specific antigen. So T cells with an intermediate phenotype or intermediate affinity for self-antigen, they upregulate a specific transcription factor called FOXP3, and they become regulatory T cells. And so FOXP3 is a transcription factor that really locks the phenotype of these T Regs in place. This is our basic outline for bringing therapeutics into the clinic. So my lab is in charge of the early part. We take from the inception through the in vivo proof of principle testing, and then we transition the expansion and purification expansion protocols over to the GMP lab for clinical scale-up and to initiate the clinical trials. So my lab doesn't have to worry about actually growing the products that are used in the patients themselves. However, we do monitor the phenotype and the in vitro function of the T Reg products. And with the help of the translational therapy lab, we monitor the T Reg phenotype and number in patient samples as well. So we'll talk about that in a little bit. Okay, so to develop thymic T Reg for clinical trials, many of our stem cell transplants at the University of Minnesota receive their stem cells from umbilical cord blood. And so we initially focused our efforts at expanding T Reg from umbilical cord blood, and as I'll show you later, this proved very fortuitous. So T Reg are a very minor population of T cells, comprising, only 5% of total T cells, and T cells are only 30% of total cells in the cord blood unit as well. But they can be purified based on their expression of CD25, and CD25 is the high affinity subunit of the IL-2 receptor. And so not only is it going to be important for purification of these cells, but IL-2 will also be critical for expanding these cells in vitro. And because of these low cell numbers, directly after purification, it required an in vitro expansion step in order to use them in clinical trials. And the fact that they express the high affinity receptor for IL-2 helped their expansion. So after many attempts, we finally developed a protocol to expand these T Regs, and that protocol was able to maintain in vitro suppressive function. So to test in vivo efficacy and provide proof of principle, we used a xenogenetic model of GVHD in which human PBMC are injected into an immunodeficient animal. And in a xenoreactive setting then, these mice develop a graft versus host disease like disease. and then we can follow the pathology, and one way to measure it is obviously through death. And if we co-inject regulatory T cells at the same time as the PBMC, we can greatly expand survival. So this was our proof of concept that regulatory T cells might work in human patients as well. And so this led to our first clinical trial, which was headed by Claudio Brunstein from HemOnc. And first, the schema for this trial was that T Regs were initially purified from a separate umbilical cord blood unit. They were expanded with CD328 beads to activate the regulatory T cells, again grown in high-dose IL-2, and we could get expansion of these cells up to about a total of 4 billion cells. These expanded T Regs were then injected into hosts that were receiving a double umbilical cord blood transplant, and after transplantation of the T Regs, we monitored the T Reg number and phenotype in the patients. So even though this was just a safety study, patients who received 3 million T Regs per kilogram had a significantly reduced incidence of severe GVHD. So we definitely had an effect. But because of low T Reg yield, we were not able to meet this dose of 3 million per kilogram in several of our patients, nor were we able to try and dose escalate up from 3 kilograms or 3 million cells per kilogram to see if we could increase efficacy. Therefore, using our standard model, the protocol came back to the basic research lab so that we could try and improve expansion and improve the T Regs as a therapeutic. Right. So actually, this model, the proof of concept model, was helpful in many different ways, and I kind of glossed over the fact that first by showing that the T Regs didn't make disease worse, that again, in our clinic or in any preparation of regulatory T cells, there's always some contamination from non-T regulatory cells. And so much of the world was concerned with what would those cells do when transferred into the patient too. So first, this experiment showed that the contaminating cells wouldn't make disease worse. In addition, we enhanced survival. So, first the model showed that we have safety. And second, what I am not presenting here is we looked at a number of different ratios of regulatory T cells to PBMC. And for this experiment, we found that the optimum was really a 1-to-1 ratio, or 1 T Reg per PBMC. And again, since the 30% of PBMC are T cells, the ratio is 3-to-1. So we were able to develop an idea of how many T Regs would likely be required for efficacy. And so the 3 million per kilogram that we were able to achieve in our clinical trial was below what we knew or what we expected to be optimal for efficacy in patients as well. So that really, was good that we could drive expansion up to the levels to achieve 3 million per kilogram, but we'd really hoped to go much higher. And so again, the idea then is that the expansion came back to the basic research lab so we could try and further expand these cells. So the model the mechanism that we had used to expand the cells initially were these CD328 beads. And again, just more information about them. They're a bead to which antibodies to CD3, which is part of the T cell receptor, and CD28 are attached. And so these beads are able to engage the two major receptors that drive T cell proliferation, TCR and CD28. And so we switched from these beads to an artificial antigen presenting cell in which K562 cells were engineered by lentivirus to express CD64, which is the high affinity FC receptor, and CD86, which is the natural ligand for CD28. And so these artificial antigen presenting cells, we loaded with the same the same antibody to the T cell receptor as the beads had, and then the CD86, the cells also express CD86. So by using these artificial antigen presenting cells, we were able to increase expansion of our T Reg by tenfold. And then we also found that we could re-stimulate the umbilical cord blood T Reg with the artificial antigen presenting cells. And re-stimulation of these cells, we didn't get loss of purity or suppressive function. And that could drive expansion an additional 15-fold. So in total, we were able to increase expansion over 150-fold. So by incorporating these two steps into our clinical trial, the overall clinical trial didn't change because we were able to keep the two stimulations in the same time frame as the original trial, which was a 17-day expansion, and now even with re-stimulation, expansion is just a 19-day expansion. So not much changed. Again, the T Reg from a single umbilical cord blood unit expanded in vitro. Oh, I forgot to change this number. With artificial antigen presenting cells instead of CD28 beads, and the T Regs were infused into double cord blood, patients receiving a double cord blood transplant. And this expansion protocol allowed us to achieve doses 30-fold higher than what we achieved in the first group of patients that used the anti-CD328 beads. So we were able to treat 12 patients with a dose as high as 100 million per kilogram. And so using our xenogenic model in the basic research experiments, we found that the number of T Regs in circulation correlated with survival. So the higher number of regulatory T cells we could have in the mouse, there was a direct correlation with how long the animals would survive. So with that in mind, we also wanted to look at the number of regulatory T cells that were present in the patients receiving these transplants. And so in the first cohort of patients receiving 3 million cells per kilogram, we were able to see the T Regs, but the total number was much lower. So when we bumped up the T Reg dose to 10 million or 30 million or 100 million per kilogram, we drove a great increase in the number of T Regs that were present in the patients. And just as kind of an aside, by day 15, the T Regs were completely absent from the patient, and initially it was somewhat worrisome because we were worried that would correlate with decreased efficacy. But as you'll see, the T Regs still do their job. So it appears that the T Regs are injected, they inhibit the initial expansion of of these alloreactive T cell clones, and then they're no longer needed. And that actually turns out to be helpful because another thing that we were worried about is, if you have this large bolus of regulatory T cells that can suppress all immune activation, that might cause the patients to die from infectious pathogens and whatnot because there'd be a general immunosuppression. So in the end, this absence of T Regs at the later time point might prove to be helpful. Okay, so again, this was the first cohort of patients, again, that had significantly reduced GVHD. Now, with our second cohort of patients, we found that, thanks. Thanks. There we go. So with our second cohort of patients, only one person developed graft versus host disease. 1 of 12 patients developed graft versus host disease, which was a great success. And John, I think that one person who developed GVHD, it was an odd GVHD as well, right? Yeah, so now this therapeutic seems to be working very well. And we can reproducibly generate the number of cells required. So in addition to our cord blood Treg therapeutic, we wanted to generate a Treg therapeutic that would work for peripheral blood stem cell transplants as well, particularly for matched related donor stem cell transplantation. And so the benefits from this type of Treg therapeutic would be that the stem cells and Treg come from the same donor, and so there wouldn't be an alloreaction against the adoptively transferred T cells and T Regs. In addition, we'd be able to achieve a higher dose because the starting cell number is going to be much higher. So we can purify 150 million T Regs from a peripheral blood apheresis unit versus 4 to 7 million for a cord blood unit. And then this would also serve as a template for other Treg applications, like using Tregs for organ transplantation or autoimmune disease, situations where you wouldn't use a umbilical cord blood unit. So, unlike the situation in cord blood, where you had a more relatively more defined CD25 bright population that could be purified to fairly high homogeneity. When we looked at peripheral blood, you had this large CD25 intermediate population that is actually activated T cells, kind of exactly what we don't want. And even after purification, these cells are still present. And so in order to develop the peripheral blood Treg as a therapeutic, we had to expand these Tregs in the presence of rapamycin, which inhibited the expansion of the effector T cells. And it's an easy one-line statement at this presentation, but of course it took much longer than that in the lab. And so again, the general overview for the path that we used to bring peripheral blood Tregs to the clinic. As I was saying, we had to figure out the purification and the expansion conditions with rapamycin. And then we went through the proof of principle again, showing that these peripheral blood regulatory T cells suppress graft versus host disease in our xenogenetic model of GVHD, and again, did not make disease worse, even though there were some contaminating T cells. And so then we translated the protocol to the GMP lab and started the clinical trial. And this clinical trial was headed by Dr. McMillan and Peds BMT. And the general outline was that a non-mobilized apheresis product, Tregs were purified from the non-mobilized apheresis product. Again, in high number, we induced the proliferation with anti-CD328 beads because this was done kind of pre-KT6486 era, expanded them in high-dose IL-2 with rapamycin, and were able to generate a very large number of T Regs. And we were able to infuse them again with a stem cell transplant from a matched-related donor. Again, it was a day 18 mobilized peripheral blood apheresis, and it was from the same donor as the regulatory T cells. So the results from this trial, from kind of the manufacturing end, all the peripheral blood Treg products met lot release criteria. They were suppressive in vitro, and we were able to achieve doses of up to 100 million per kilogram. Now the downside was that we were only able to accumulate three patients. So this very slow accumulation was likely due to the fact that the donors had to give 2 apheresis donations, and they had to stick around for that 18 days. So what we found was that peripheral blood Treg cellular therapy was safe. Again, these contaminating T cells that were present in the Treg preparation didn't cause further disease. And again, we were achieving doses up to 100 million per kilogram. So in some of these patients, we were administering 10 billion cells, and if even 10% were non-Tregs, that's a billion cells that were non-Tregs. That's a large number, and so the fact that the Treg cellular therapy was safe was still a big deal. So the future directions that we'd like to take the thymic Treg cellular therapy, first we'd like to develop umbilical cord blood Treg banks so that we could treat multiple patients. Right now it's one Treg product per patient. It's not very efficient on both a cost effectiveness sense, but also there's a lot of product to product variability. And so it would be, now that we can get these large scale expansions, it would be really nice to develop one product that we know has high efficacy and then treat multiple patients. And we have experiments showing that Treg matching to the donor T cells is not important. So it's not as though we would need to make an unlimited number of bank cells to match even 4 out of 6 HLA alleles. So a limited number of banks could treat the majority of patients. And then the second thing we'd like to do is to develop Treg cellular therapy for treatment of acute and chronic GVHD. And so the best option for that would be to have a product that could be directly thawed and injected at the time that patients were developing graft versus host disease. So we did some research in the lab to see if that was a possibility. And again, looking at our xenogenetic model of GVHD, we found that, so the black line is PBMC only, so that's the baseline disease. And so Tregs that were freshly thawed and injected were really no longer effective at suppressing graft versus host disease. However, if we incubated them overnight in high-dose IL-2, they regained their suppressive function. So there's hope for this therapy. Now, obviously, we don't want patients who've been diagnosed with graft versus host disease to wait overnight with no treatment. So we still need to improve upon that. But it looks like we will be able to develop a treatment option as well. So when I teach immunology students, I explain that Tregs are capable of such high rates of expansion that if unchecked, you'd end up with about 72 pounds of E. coli specific T cells in 3 weeks. So obviously there must be a system of checks and balances, and not surprisingly, that system of checks and balances involves regulatory T cells. And these regulatory T cells are used to limit immune activation in response to chronic pathogens. So kind of this is the outline, right? So what we were talking about earlier is that these regulatory T cells that have intermediate affinity for self-antigens are selected in the thymus and then come out to the periphery where they're able to regulate effector cells or become effector T Reg cells and are able to work on activated T cells. But in the case of the regulatory T cells that arise in response to chronic stimulation, they come from the thymus as just regular naive cells. And then when they're activated in response to specific stimuli, examples are TGF-beta, IL-10, or retinoic acid, they are induced to become a regulatory T cell and then are able to impact other antigen-specific T cells. And so there's been a lot of news lately about the positive role of gut microbiota in maintaining health. And this is likely one of the things that we've kind of discovered in the last year or so, is that this is likely due in part because they're able to induce regulatory T cells. So commensal bacteria in the gut produce metabolites and help metabolize and activate retinoic acid, and the combination of these two are another signal that induces regulatory T cells. So we found that a combination of rapamycin and TGF-beta was able to induce suppressive function by expanding we found that we could induce suppressive function in T cells by expanding them in the presence of rapamycin and TGF-beta, and these rapa TGF-beta induced T Regs were as potent as thymic T Regs in our xenogenetic model of GVHD. The animals receiving PBMC only succumb rather rapidly to GVHD. Animals receiving in vitro or adoptive transfer of regulatory T cells with their PBMC had enhanced survival, and now these adoptive transfer of the rapid TGF-beta induced T Regs also significantly enhanced survival. So they were as potent as thymic regulatory T cells, and their yield, because we were able to start with total T cells from an apheresis product. So rather than starting with 7 million cells from a cord blood unit, we were able to start with 3 billion cells from an apheresis unit. And then after expansion, that first clinical trial that we had with cord blood was only able to generate 4 billion cells. And with induced regulatory T cells, we were able to generate 237 billion cells. So the schema for this trial was very similar to the peripheral blood T Reg trial. It was headed again by Dr. McMillan. Total T cells were purified from a peripheral blood apheresis unit. They were expanded in the presence now with these antigen or the artificial antigen presenting cells, these KT6486 cells, in high-dose IL-2 with rapamycin and TGF-beta. We're able to generate high numbers of cells, and they were transplanted into patients receiving a matched-related donor stem cell transplant. The results from this trial, which is just concluding now, again, if we look at the T Reg products, so all the T Reg products met release criteria. Again, we were able to achieve doses of up to 100 million per kilogram, and all of the induced T Reg products were suppressive in vitro. Again, the first do no harm. So the patients receiving the T-Reg cellular therapy, didn't have any adverse reactions, so the therapy itself was safe. Unfortunately, to this point, we have not seen any therapeutic benefit in the patients that have received these regulatory T cells. And one concerning thing that we have noted anyway when looking at the cells from the patients was that it appears that these adoptively transferred induced T Regs lose T Reg specific markers, FOXP3 and CD25. And so perhaps they also lose their suppressive function. But importantly, even though they lost these markers, they didn't exacerbate graft versus host disease. So now that so I'm going to kind of try and bring in a little bit of basic biology now. Yeah, Tony. So the important point is that we really only have to provide immunosuppression, and I really should have gone through this too. We only have to provide immunosuppression during the period of time before the stem cells seed the thymus, and so once the stem cells seed the thymus, they grow up in that host and no longer are able to induce graft versus host disease. So we just have to cover it for that little period of time, and and in thinking of how we might translate this to autoimmunity, obviously that's going to be a little bit different because, whatever is inducing the autoimmune disease is probably around all the time. So that may require, multiple injections or something like that. But for this specific use, T Regs are kind of the perfect therapy. They go in, they do their job, and they don't, then the patient takes over tolerance themselves. Any other kind of clinically related questions? Yeah, Joe. Yeah, so, yeah, good. So the first part was would how would our T Reg therapy impact, or what would the impact be on immunosuppressant drugs? And I think the goal would certainly be that the patients would be able to get off cyclosporin or rapamycin or FK506, and I guess kind of if I can borrow from some basic research that's been done in autoimmune disease, so the idea of graft rejection. So in the situation where you have graft rejection, the T Regs can go induce tolerance, and then normally you can't take the graft recipient patients off of immune suppressive drugs. But if you treat them with T Regs, you were able to take them off FK506. And so I think, John, that's right? I mean, the hope is that we would be able to not worry about other mechanisms of immunosuppression. And then kind of the flip side to that is this question of global immunosuppression. And so, what's really driving this is that if you take a mouse and you transduce all T cells with FOXP3, so they're nearly all regulatory T cells. Now these mice are susceptible to commensal bacteria and other opportunistic infections, and actually by by enhancing suppressive function, you actually make the health of the animals worse. So again, we're transferring in, 10 to 30 billion of these T Regs, right? So it's a huge number, and there was a great worry that you would get global immunosuppression, and the patients then would, be at more risk to opportunistic infections. And so that's something that Claudio and Margie have analyzed in their patients, and thus far there's been no increased risk of infection with the patients that have received the T Reg therapy. That's what it was.
So it was a concern, but it's not yet happened. Perhaps because we're giving T Regs in the full, but not.
All right. Well, we can engage somebody else for help with answering some of those. So I just wanted to take again, my work is in the basic science of regulatory T cells, and so I just wanted to kind of give a quick overview of an example of some of the basic science work that we're doing to try and develop the next T Reg therapeutic. One of the things that we've done is we've looked by miRNA microarray at differential expression of these miRNA on T effector cells versus T Regs. So microRNA are small 22 nucleotide mRNA molecules, miRNA molecules that bind to specific mRNA species and regulate transcription of these mRNA molecules, regulates both stability and transcription. So again, we did a microRNA, an miRNA, microRNA, comparing T effector cells and regulatory T cells, and obviously there were, out of 5,000 species of miRNA molecules, there were many that were differentially expressed, some that were more highly expressed in T Regs, and so to look and see if microRNA species are having an effect on T Regs, we use this integumer technology, and so integumers are just anti-miRNA molecules, and once you get them across the cell membrane, they sequester the miRNA molecules and inhibit their action. So one of the miRNA molecules that was specifically expressed in T Regs was something called MIR146B, and that's known to block TRAF6 expression, and TRAF6 is involved in NF-kappa B activation and T cell expansion. So with no treatment, T Regs express very little TRAF6, but with this integumer treatment, we upregulate TRAF6 expression, and upregulating TRAF6 expression increases T Reg expansion. And now when we look at T Reg efficacy in our in vivo model of GVHD, we see untreated or PBMC only animals, T Reg plus PBMC animals, T Reg treated with scrambled RNA, and then we see increased efficacy of these T Regs that were treated with integumer. So again, we do all these experiments with GMP compliant reagents. We work with some of the top people in the field who are developing therapeutics. So this is a protocol now that would be able to be translated to the GMP facility. But I guess first regulatory T cell therapy has to be, how far does it have to go, John? Does it have to be, do we have to get to phase 3 or something like that? When can we start making these these changes. Because, of course, as a basic scientist, I develop these things, and I just think, okay, the next patient that we treat, we should just do this treatment or that treatment to the T Regs, and it's not quite that way. Yeah. So again, we've developed a number of different mechanisms to improve the T Reg efficacy in our in vivo model of GVHD, and when appropriate, we can incorporate that for actual clinical trials. So with that, I'd like to thank the people in the lab, Sophia Haney, Kyle Smith, Sangya Singh, and Bailey Hoyle, and MCT, the people who work at the MCT facility who've really helped, Dave McKenna, Diane Cadidlo, and Darren Sumstead. Claudio Brunstein, again, was in charge of the UCB T-Reg trials, and Margie McMillan was in charge of the PB T-Reg and IT-Reg clinical trials. And our collaborators at the University of Pennsylvania, Carl June, Jim Riley, and Bruce Levine, who really developed these artificial antigen presenting cells that have, kind of really launched this regulatory T-cell therapy into a new higher level, since we can generate so many more cells. So with that, I'll take any other questions. Yeah, that is my thought. And so that leads kind of into oh, right. So the question was, what do I think leads to the collapse of the regulatory T cells that are going into the patients? And the suggestion was IL-2, and I would absolutely agree with that. So in our cultures, they're receiving very high doses of IL-2, and it's not unusual for cells to get kind of addicted to cytokines. And so IL-2 is probably the culprit. And so in thinking that, there have been other clinical trials where IL-2 was given, exogenous IL-2 was given low dose or ultra low dose IL-2. And we did try that in our xenogenetic model, and I guess Claudio did try that with a number of his patients as well, but it was not efficacious for us at all to yeah, I didn't. And I'm kind of glad that you put it that way. I hesitated to put it so strongly, but in our xenogenetic model, it was really not tolerated either. So, Tony.
And there's a study in IPD's population, not with T regulatory cells,
yeah.
where in diabetes, rapamycin actually increased activated T cells and accelerated loss of beta cells. So the diabetes community is really nervous about rapamycin.
Well, I was not aware of that. And even conceptually, that's kind of hard to digest, too.
So one other aspect of this is the practical aspect of all this. I mean, just to bring up a couple of points, because this is what you did, and that is that, when we talk about the KT, So from a practical point of view, as you heard, we used the KT or the K562 cell line that was genetically modified as a way of expanding the T-reg population in the laboratory before injecting it. However, K562 is an erythroleukemia cell line. So what did the FDA ask you to do in anticipation of you giving, at the same time giving T-reg, we're also giving potentially erythroleukemia to the patients?
Yeah, so these KT cells are irradiated with 10,000 rads, and if we irradiate the cells, and specifically the cells that we use to expand the clinical products, if you put them back into culture, no cells grow out. But there have been other trials where transformed cells have been irradiated and given to patients, and evidently they weren't all dead, and they actually did cause tumors. And so we kind of had to prove that these KT cells were indeed dead, and we couldn't find them in the cultures that would go into patients. And you know, one difference between those studies and our study is that it was, I can't remember exactly what it was, but it was like a cell had been transduced to express human growth factor or something like that, and those cells themselves were given to the patients. For us, we're just treating our T Regs and then expanding them in vitro for 21 days. And so if the T Reg, or if the KT cells weren't dead, you would see them continue to grow in the presence of the T Regs. And so as part of the, what do you call it? The cells have to pass, before they can be injected into patients, yeah, thanks. They have to pass lot release. And so the idea then is that we would be able to detect a KT expansion there. But we had to go back through, all the patients that we had treated and kind of some basic science stuff and show that the KT cells were not expanding in these cultures at all.
So another practical thing is that each one of these products costs $100,000. So every single patient, the product manufacturer was on the order of $100,000. And so it gives you an idea of the expense. But one of the things that Keli described at the end was that if we create these large banks of cells, even though the cost of a single manufacturer may be high, we can probably, employ those in dozens of patients, and that obviously reduces the cost per patient. So we have practical issues as well as the science behind it that we have to deal with. And sometimes the practical issues, deep six the whole project. As you gave an example of the NT reg in the peripheral blood stem cell transplant patients, it was not practical for the donor to come twice. Once to give the peripheral blood for creation of the regulatory T cell, and the second time to give the hematopoietic stem cell graft. And so practical issues can play a major role in how effective the treatment plan is actually. Yes.
Yeah, Joe.
Where is this going in terms of solid organ transplant?
Yeah. So
where is it going in terms of solid organ transplant or other types of
so here at the University of Minnesota, Eric Finger, who's in the Department of Surgery, is trying to develop a T Reg therapy for lung liver transplants. And he's having more difficulty than we had getting it off the ground. And again, I guess my thought is that I don't want to say that. So we're trying to get it into practice for solid organ transplantation here, and other organ transplantation uses. I'm not aware of other clinical trials that are actively going for.
Well, they're being considered for auto islets, or I should not.
Yeah,
so islet transplants and.
And an important point is that they are in use for treatment of or of diabetes, right? Recent onset diabetes. Jeff Bluestone at Stanford is using UCSF, is using regulatory in vitro expanded regulatory T cells to treat patients with recent onset diabetes successfully. Yeah. Right. I guess, thank you.
Any other questions?
Lydia.
Joe's question about the solid organ transplant. I don't know if one of the differences as to why it's been more difficult to develop them for solid organ is that in solid organ, they're on immune suppression for the rest of their life. And especially early on, they get like in the kidney transplants, they get thymoglobulin and then CellCept or mycophenolic acid and then tacrolimus or FK506. So that I think is going to make it a little harder for the solid organ because they're on for life.
It's kind of the same thing that we're facing with treatment options for GVHD, right? I mean, the immunosuppression that's used for solid organ transplantation affects the T Regs, right? I mean, they don't like to expand in the presence of their IL-2 dependence, so inhibiting calcineurin really impacts their ability to survive in vitro. And, you know, with solid organ transplantation, one of the things that we, Dr. Finger faces anyway, is that they want an autologous T Reg product, right? And so there's just, you know, it's difficult to identify a donor, to, you know, expand the regulatory T cells and have them ready at the time of transplant, you know, when you would give the immunosuppression. So, yeah, I think in the end, graft versus host disease, prevention of graft versus host disease was really the perfect place to start with T Reg therapy. Thanks.
You have been disconnected from the meeting.